Tissue Regeneration, Chapter 22 of the 12th edition, Part 1 of 2. Chapter opener looks at planarian and its remarkable ability to regenerate. This was a planarian that was surgically acted on, and you can see that it has 4 separate heads now because of the way that one of them was split and the regeneration occurred. There are a number of different organisms that have different capabilities of regeneration. So representative organisms and their comparative regenerative capabilities are shown here. We have Hydra, which you can have an entire individual regenerate from just a small fragment. Planaria, you can have a whole anterior or posterior half of the body regenerate if you cut off the other half. Amphibians and fish, faux limbs, tails, fins, limbs, central nervous system, heart, and other organs can regenerate in these representative organisms. Mammals have the most limited capability for regeneration. Deer can regenerate their antlers, and mice and men can regenerate some organs. There are 4 major types of regeneration. The first is called stem cell mediated regeneration. Stem cells mediate regrowth of certain organs and tissues, including skin, hair, and blood. An example organism that we're going to look at is planaria. Epimorphosis is another type of regeneration. This is where adult structures can de-differentiate and re-differentiate to form a new structure. For example, amphibian limbs undergo epimorphosis. Examples include salamander. Morphollaxis is another type of regeneration. Here we get repatterning of existing tissue. And the example we're going to look at is Hydra. And fourth, compensatory regeneration. This is where differentiated cells divide, and our example tissue is the mammalian liver. There are pictures of the 4 different types of regeneration that I'll be showing to remind you of them as we go along with the different examples. We have stem cell mediated regeneration involving a multipotent stem cell. We have epimorphosis that does not involve a stem cell. Compensatory regulation has a different kind of stem cell because it's a differentiated cell. And then morphollaxis is shown here, and we'll talk about that when we get to that. I am not going to play this video for you. Please go to this link and look at the HHMI video. It's free streaming. It's about 12 minutes long. And planarian is an example of stem cell mediated regeneration. Stem cell mediated regeneration is pictured here. It involves a multipotent stem cell. Multipotent stem cell, meaning it's not pluripotent or totipotent. It can't give rise to all tissues and extra embryonic tissues. It can't even give rise to all of the cells in a body, but it has the capability of regenerating multiple different types of cells, including shown here, a neuron, a muscle cell, and a fibroblast. These, you can also have examples of organisms that are going to have unipotent stem cells that can stem cells means they can self-renew, as well as give rise to a cell that's going to differentiate. In planaria, what we have is the ability to regenerate is accomplished by a pluripotent stem cell population of neoblasts. So this one's actually pluripotent. Clonogenic neuroblasts is their name, and they are shown here in this figure A. We have them stained red. So everywhere you see the red fluorescent staining, that means there is a clonogenic neuroblast cell there, and they can generate a colony of cells that can regrow organs. You can see that they're located in most of the body. They are absent from the head and specifically the eye spot area. So below the eye spots we have them, but in this area that is called the pharynx that you'll learn about on the HHMI video that you'll be viewing, also does not have these clonogenic stem cells. Now what you can do is radiate. Expose this poor thing to radiation. If you give it a low dose of radiation, 1,750 rads, you can eliminate most of their clonogenic neuroblast cells, but not all of them. And as long as you have at least one left, the organism is capable of regenerating all these different cell types. However, if you give it a higher dose of radiation, 6,000 rad, then in order for regeneration to occur, you're going to have to transplant a clonogenic neuroblast stem cell in here. And it has the capability of regenerating all of those stem cells so that the planarian can regain its full capacity to regenerate when cut in half or thirds or so forth. So planarian flatworm regeneration involves pluripotent stem cells called clonogenic neuroblasts or neoblasts. Irradiation that kills all the clonogenic neoblasts blocks regeneration and ultimately leads to the death of the flatworm. One transplanted neoblast into an irradiated donor can completely restore all of the cells in the flatworm. All it takes is one. The secret to success for the planarian is our friend Wnt. And we learned about Wnt as a morphogen. It's a paracrine factor. Expression of it is going to dictate the planarian regeneration. And so I want to give you a little reminder of the Wnt pathway. So Wnt is a ligand. So when there is no Wnt present, there are no signal transductions, and the target, the cellular target, beta-catenin, is degraded. So in the absence of Wnt, beta-catenin transcription factor is degraded by the proteasome. When Wnt ligand is present, the receptors such as LGR, R-respondin, and disheveled can interact with Wnt and to signal transduction that will prevent beta-catenin from being destroyed, and instead it is translocated into the nucleus where it can interact with some of its cofactor proteins and start transcription of whatever genes need to be made in order to regenerate the body parts. So how do flatworms establish their anterior-posterior axis? Remember, anterior is the head and posterior is the tail. Well, it's a Wnt gradient. So up here at the head, we don't want Wnt, no Wnt inhibition. At the tail, we do want Wnt activity. So if you cut this thing into thirds and you leave only the torso where it has all those clonogenic stem cells, it's going to allow the head to grow here as long as Wnt is absent, and the tail to grow here as long as Wnt is present. So what do you predict will happen if we completely lost Wnt activity? Let's say we had a planarian that was mutant, that had a double mutant in Wnt, and so there was no Wnt ligand. What do you think is going to happen? Now going back to this chart, in the absence of Wnt, what happens? We get heads, okay? So let's say if we're going to cut this thing in thirds again, and there's no Wnt being expressed, what's going to happen is we're going to get heads growing on both sides. And so we're going to have a two-headed planarian. What do you think would happen if the opposite were true? If you had overactive Wnt, what would you predict? Now remember, you're going to need a lot of Wnt to make tails. So if you cut this thing into thirds again, what do you think is going to happen? If you guessed it's going to have two tails, you are correct, because tails grow when Wnt is active. And these studies were actually done and published by Tanaka and Gilbert in Nature Cell Biology in 2008. Along with their publication, they showed that there is a polarity in planarial regeneration, and they gave us a little map as to how that needs to happen. So here's a normal planarian here. We've got the head, we've got the tail. Here are two abnormal planarians shown in panel B and panel C. We have beta-catenin blocked in panel B, and Wnt1 is blocked in panel C. And it's harder to see in panel B, but it's got two heads. There are two eye spots on either side of the creature, just like in the Wnt1. And that just reflects the Wnt pathway. Beta-catenin is a target of the Wnt pathway and is the thing that goes into the nucleus and transcribes genes to allow regeneration of tissue. And Wnt is the signal for that transcription. So this little map right here in panel D shows you that in the tail there is a high concentration of Wnt beta-catenin, as shown by the dark pink. As we migrate into the torso, we have less and less Wnt. And Wnt is going to stop the expression of ERK, and ERK is needed in the head. So this is reminiscent of what we studied with Drosophila, anterior, posterior, except we had bicoid and nanos that we were looking at, and caudal. So we had different proteins, but same sort of thing. We have morphogens called ERK for head development in the planarian. In other words, the absence of Wnt. And also we have the presence of Wnt in the tail and the absence of ERK in the tail. So the next type of regeneration is called epimorphous. And I'll play this animation, this little thing, if I can get to it, because it's real short.
Now, we're going to have a look at the way in which an amputated limb grows over the course of about 90 days in a salamander. That's a time-lapse movie, watching that thing grow. And now we're going to see what's really going on. Here's the salamander, and it's got a completely new limb. It looks perfect. It has inside bone, it has nerves and muscle, and it can even wiggle. In fact, it's perfect. Now we're going to cut it off. But here's the good news. It grows back. So don't be too scared. First, wound healing. Did you see that wound heal? Now we're watching what the nuke can do that we cannot do. Cells are streaming out of the surrounding tissues into the area of the wound and forming what we call a blastema, which is a group of undifferentiated cells that are in fact really just like stem cells. And they're multicolored because they come from skin, from muscle, and even from cartilage. And these have a miraculous memory of what they used to be.
And they're going to differentiate back into those things that they used to be, is what we find. But we're going to show you the evidence for that. So epimorphosis is the way of regeneration in which we have a somatic cell, a completely differentiated cell, that undergoes de-differentiation. It doesn't become a stem cell, but it's less differentiated than it was. And then it has the capability of re-differentiating into different types of tissue. This is what was shown graphically in the HHMI video. This is a figure from a paper where they cut the salamander's arm off or limb off at two different locations. One is called the proximal amputation, which is closer to the body than the distal amputation, which is further away or more distant from the body. And in both cases, you get full restoration of the limb within a matter of days. This is the organism that is being used in these experiments. It's called the spotted salamander, the Ambystoma maculatum, and here it is in its larval form. So this is some figures starring that spotted salamander, regeneration of the larval full limb. So they're looking at the larva. They're not looking at the adult here. And so they cut the limb off, and they look at it 2 days post-amputation, and you can see, just like in the video, we've got an epidermis that has reformed, and the humerus bone is right here, and the muscle cell is indicated by this M. The very top of the amputation area is going to have some tissue form there that's called the AEC. AEC stands for apical epidermal cap, and it secretes paracrine factors. So we're going to get a lot of paracrine factors, and we're going to form this blastema that she referred to in the video. Blastema is undifferentiated cells. And you can see that these cellular area increases as the days pass by. And by day 9, we have evidence that from the humerus, in panel D, we've got now the ulna and the radius forming, as indicated by the U and the R here in the humerus. And there's where the AEC is located. And then in addition to the radius and the ulna, we now get the corporal bones formed, and then the digits, digit 1 and digit 2. How does dedifferentiation occur? Well, the simple answer is changes in gene expression. Muscle-specific genes, tissue-specific genes, genes that are needed for a differentiated tissue, are downregulated, and these include MRF4 and MIF5. In contrast, proliferative genes are elevated. The expression of those are elevated. An example of that would be a gene called MSX1. Now I want to note that usually only the stem or stem-like cells undergo proliferation. Differentiated cells typically do not. So here's a question. Do the cells have a memory of what they had been? Are they dedifferentiated cells? Are these dedifferentiated cells found in the blastema? Are they multipotent? Or are they unipotent? Can they become multiple cell types or only one cell type? So just a reminder, multipotent can in any of the blastema become a muscle cell, any of those, or unipotent can only old muscle cells give rise to new muscle cells. And in other words, can a cartilage cell give rise to a muscle cell, or does it have to be an old muscle cell that creates a new muscle cell? Can you think of an experiment to differentiate between the two possibilities? Your hint is that you already know about enhancers in the green fluorescent protein. So we can take this protein from jellyfish, the green fluorescent protein, the gene that codes for it, put it behind a region in the salamander that codes, that would turn on a muscle cell, an enhancer DNA element that would turn on muscle cell expression or cartilage cell expression or the skin cell expression, and put that piece of DNA in front of a piece of DNA that codes for the green fluorescent protein. So that's exactly what was done, and it was done in this salamander, the axolotl salamander, as a model organism. It too is a vertebrate. It too is easy to rear in the lab. And it's the only salamander that really breeds multiple times a year, so you're going to have a larger number of these to work with in a given time period than maybe the spotted salamander. They're also large in size, so this makes it easier to conduct surgeries and transplants on. And so the conclusion you can see right here in the figure title is that blastema cells retain their specification even though they dedifferentiate. So they did the experiment where they created a organism that has the green fluorescent protein expressed only in the cartilage. And they took a piece of that cartilage and they transplanted it to a wild type organism. And then they cut off its limb right there at the and there a blastema formed. And then they wanted to ask, what kind of cells do we get out of there? And so the only ones, remember, that are labeled green are the cartilage cells. And yet we have to regenerate skin cells and muscle cells here But if you look at the panel B down here, there's only a small area of green. The entire blastema is not green. It's just a section of the blastema that is green, and that represents those cartilage cells. So this is evidence that cartilage cells, new cartilage cells, can only come from old cartilage cells. So these old cartilage cells de-differentiated, and then they're re-differentiated back into cartilage cells. They did not become muscle cells. And just to confirm that, they have another picture of a microscopic picture of this. Blastema cells retain their specification even though they de-differentiate, so they are called restricted progenitor cells, not stem cells. They are showing that the muscle cells here that are stained kind of an orangish color don't have any green in them whatsoever, so they did not become, we didn't get muscle cells from cartilage cells. The cartilage cells, the old cartilage cells, de-differentiated and only created new cartilage cells here. And you can see that there are these other skin cells that are dyed blue that also did not come from the green cartilage cells. So this reconfirms that the blastema cells retain their specification if they de-differentiate, but only so far. They don't go back to the stem cell level, and so they really have more of a unipotent ability to regenerate. So you can only get new cartilage cells from de-differentiated cartilage cells, old cartilage cells, and so forth. Blastema cells of axolotl salamander retain their specification. They are not multipotent stem cells, and they have a they are called restricted progenitor cells. What is required for the proliferation of blastema cells? Well, the apical ectodermal cap, or AEC, secretes a paracrine factor called FGF8, fibroblast growth factor 8. Now, we learned about this as one of the morphogens, or paracrine factors that are important for development. That's one factor. Another factor is coming from the nerves, and it was believed that it was a growth factor that was being released from these nerve cells. And they had a candidate, which is called NAG, or nute anterior gradient protein. And so they tested this by cutting some nute limbs off. So regeneration of nute limbs depends on nute anterior gradient protein. That's their conclusion. Here's the experiment they did that proves that. And this is normally supplied by the limb nerves. So that explains why they did what they did. So they took a newt and they cut both its arms off. And one of them, they also removed the nerve. And you can see here in the control panel that the left arm was regenerated and the right arm was not. And the star indicates that that's where the nerve ends, that they paired it back to that portion. So that was 7 days that both were amputated, and then they, 5 days later, put in this secret ingredient, this paracrine factor called NAG, or nute anterior gradient protein. How did they get those in the cells? Well, they had to use a little electric shock to do that, and that process is called electroporation. And when they did that, they were able to regenerate the limb, and you can see it's a little bit behind this other one that already had that growth factor before electroporation. So yeah, he's not going to have a nerve in there to move those things around and everything, but he does have, they did get that factor there, so he's going to get a new arm. So that's intermission, and we're going to be looking at the next two types of regeneration, morphollaxis and compensatory regeneration. I'm laughing because it says 3 and 5 instead of 3 and 4. Ay, ay, ay.